Okay, let's start off with question 5. If you want to look at part 1 for questions 1 and 4, you can check out the video link in the description. All right, let's start off with question 5. Some forms of fertility are due to the female producing antibodies to sperm cells. Contraceptives are being developed that use these antibodies. Antibodies can be injected into a person to give immunity. Which type of immunity develops from the injection of antibodies? So because we're injecting it and we're not developing the antibodies, that would be passive. And because we're making these antibodies, they're being developed by companies, then that would mean that it is artificial. So this would be B, artificial passive. All right, part B, in the development of these contraceptives, an agglutination assay was performed. So agglutination is what happens when the antibodies bind to the antigen and it causes the particles to stick together. Okay, antibodies were mixed with sperm cells and the time taken for 100% of the sperm cells to agglutinate was recorded. Different concentrations of antibody were tested. The number of sperm cells that escaped from the sperm cell agglutinates at each concentration of antibody was also recorded. All right, part 1, each test used 2 microliters of the sperm cell suspension at a concentration of 3 × 10⁶ cells per cm cube. So according to this units here, the concentration is the number of cells divided by the volume in cm cube. What we want to do here is, because we're given microliters, we want to convert that to cm cube first. Okay, so first let's try and convert the microliters into centimeters cube. So for 1 cm cube, you actually have 1,000 microliters. So right now we have 2 microliters and we need to convert the 2 microliters into centimeters cube. So we need to divide that by 1,000 so that we get volume. You would have to do 2 divided by 1,000. That would be 0.002 microliters. And then we would just put this number into this equation here. So we have cells equals basically the concentration times by the volume. The concentration is 3 × 10⁶, and then we would times that by the volume. Okay, let's do that. 3 × 10⁶ times by 0.002. And we should get 6,000. All right. Let's go on to part 2. The graph shows the result of this assay. And then the percentage of sperm cells that escaped compared with the control with no antibodies. Okay, analyze the data to comment on these results. So let's look at the first graph. So whenever we're talking about trends, it's always good to sort of phrase your trend as something increases, this increases or decreases. So in this case, as the concentration of antibody increases, we can see that the time taken for 100% agglutination decreases. You can also talk about the lack of error bars for this graph, because the error bars show you how spread out your data is. Because whenever they do this experiment, they're doing it multiple times. They're doing repeats, right? And what we have here is just the mean. And you want error bars to basically show you how spread out your data is across the mean. For the second graph, they do have error bars. We can see here that when the concentration of antibody is more than 6.25 micrograms per centimeter cube, the percentage of sperm cells that escaped is relatively low. And we can see that because the error bars do overlap between these 1, 2, 3, 4, 5 concentrations, that there is no statistical significance between them. All right, so let's write that down. So from the first graph, see that as the antibody concentration increases, the time taken for the sperm cells to agglutinate decreases. However, because there are no error bars, we conclude, that the time taken between the concentrations statistically significant. In the second graph, we can see that an antibody concentration greater than 6.25 micrograms per cm cube, or prevents the sperm from escaping, the error bars for 6.25 to 100 micrograms per centimeter cube overlap, which means an increase in concentration does not affect the percentage of sperm escaping. Let's go through the marks. Okay, let's go through the marks. The first one is as the antibody concentration increases, the time taken for the sperm to agglutinate decreases. That's one mark. And then you can comment on the lack of error bars and how we can't really conclude that the time taken is statistically significant. So that's 2 marks so far. The third mark is basically stating that the antibodies do prevent the sperm from escaping, which I write over here. Fourth and fifth mark is because they overlap, an increase in the concentration does not affect the percentage of sperm escaping. All right, let's go to part 2. Observation of the sperm cells agglutinated by these antibodies show that some were joined head to head, some head to flagellum, and some flagellum to flagellum. Explain these observations. All right, so I've wrote out these drawings to illustrate what is happening here. So over on the left, we have two sperms that are joined head to head. In the middle, we have two sperms that are joined flagellum to flagellum. And over here, we have two sperms that are joining head to flagellum. So if you recall, when it comes to an antibody, your antibody is Y-shaped, and it has these two identical binding sites. And if you recall, antibodies can only bind to specific antigens. So in this scenario, you could have, let's say, these green antigens that are only found on the head, and therefore this green antibody can bind and recognize the antigens on the head only, and that causes head-to-head joining. Now, the red antigen is only found in the tails, and so the red antibody would then bind and recognize the antigens on the tail or on the flagellum, and it would lead to flagellum binding. And lastly, you have these blue antigens that are scattered in both the head and the flagellum region, and that would allow for the antibody to bind to, or the antibody to join the head and the flagellum together. So we can say antibodies have binding sites that allow for them to bind specifically to an antigen. There may be three types of antibodies that could allow for this agglutination for head only or flagellum only. Binding to occur. The may have recognized antigens that are only present in or only present on the head and the flagellum. For head to flagellum binding to occur may have been antibodies that recognized antigens present throughout the whole sperm, or you can say that is present in both the head and the flagellum. All right, let's go through the marks. The first mark is antibodies have binding sites. Let me highlight that here. The second mark is antibodies are specific to antigens. And then you can say there may be the same antigen on both the head and the flagellum. So this is this part here. For head and flagellum binding to occur, there must have been an antigen that's present throughout the whole sperm. And then you can talk about how for head only and flagellum only, there might be different antigens. So antigens that are only present in the head and the flagellum. And then the last mark would be there are different types of antibodies present. All right, let's go to the last part. Part C, in the development of these contraceptives, the effect of the concentration of sperm cells on the time taken to agglutinate was also investigated. Explain why agglutination can take longer at low and at high concentrations of sperm cells. Okay, so at low concentrations, it would take longer because of the infrequent collisions between the antibody and the sperm. And at high concentrations, it would take longer because there might not be enough antibodies to sort of agglutinate the sperm cells. All right, let's write that down. So at low concentrations, time taken is longer because of the lack of collisions between the sperm and the antibody. At high concentrations, it is because there may not be enough antibodies agglutinate the sperm cells. All right. So those are the 2 marks. Let's go on to question 6. Scientists have removed thylakoids from chloroplasts to make artificial chloroplasts. Part A, the diagram shows part of a chloroplast. Part 1 says draw and label 3 other structures found in the chloroplast. So I've included this diagram here from the marking scheme. So some of the features that you can include are the inner membrane and the intermembrane space. And if you recall, organelles like your chloroplast and your mitochondria both have their own 70S ribosomes and a loop of DNA. You can also include the starch grain and the lipid droplets and label the stroma, which is basically the space that we call inside of the chloroplast. And then we actually have this intergranal membrane, or sometimes you'll hear it be referred to as a lamellae, and that actually separates the granum or the stacks of thylakoids so they don't end up being stacked together, and that's to maximize light absorption. All right, let's go on to part 2. Chloroplasts can be 3 micrometers in length. Calculate the magnification of this diagram for a chloroplast that is 3 micrometers in length. Give your answer to 2 significant figures. So the equation for magnification is magnification equals image size over actual size. Here, the actual size is 3 micrometers. And if we use a ruler, let me just remove this. So if we use a ruler, we can measure the length of this chloroplast. So use a ruler to measure the length of the chloroplast, and you should get perhaps 8 centimeters, around 8 centimeters. All right, so now that we have 8 centimeters, we want to convert that into micrometers. So 8 centimeters would actually be 80 millimeters. And then to convert that into micrometers, we have to times it by 1,000 to get 80,000 micrometers. And then now we can just do 80,000, divide that by 3, which is 26666. So let's write that down. All right, let's go on to part B. Explain the importance of the thylakoid membranes in the light-dependent stage of photosynthesis. All right, so I've included this diagram here. Let me just orient you to where we are. So this is the thylakoid that we see here, and this is the lumen of the thylakoid. Let's just scroll up so you can see. So your thylakoid is here. So we have your thylakoids are stacked up on top of each other, and it's responsible for the light-dependent reaction of photosynthesis. So let's scroll back down. So in your thylakoid membrane, this is the membrane and this is the stroma, which is basically the space inside of the chloroplast. So on the membrane, we have these things called photosystem. We can see photosystem 2 over here and photosystem 1 over here. And these photosystems contain pigments, which allow for light to be absorbed. And when light is absorbed, it causes the electrons to get excited. So you can see the electrons here, they're getting excited, and then they're able to move down the electron transport chain, as you can see here. And as it goes down the electron transport chain, the energy from the electrons are used to pump up these protons into the thylakoid lumen. So eventually what you have is this buildup of hydrogen ions inside of the thylakoid lumen, right? And the only way for them to be able to get out or to leave the lumen, which they really want to do because it's very positively charged inside and there's a high concentration inside, is they want to be able to leave, and the only way they can leave is through this ATP synthase. So as the hydrogen ions go down the ATP synthase, it kind of works similar to like a water wheel, where as something flows down, it causes this reaction to occur. So it causes the formation of ATP, fumarate ADP, and inorganic phosphate. So this process is what we refer to as chemiosmosis. Let me just write that down. So chemiosmosis is a process in which ions go down their electrochemical gradient. You can also talk about how this process is an example of photophosphorylation. So photo means light, and phosphorylation refers to the generation of ATP that we see here. So it's basically using light to make ATP. All right, so I'm just going to erase this and let's write this down. So we can say the membrane contains photosystem 1 and 2, which has pigments that absorb light. This causes the electrons to become excited and move along the electron transport chain, which uses the electrons to pump, you can say protons or you can say hydrogen ions. So I'm just going to write H plus ions from the stroma to the thylakoid lumen. Again, remember, we're talking about the importance of the thylakoid membrane, right? So the membrane is there to ensure that there is a buildup of hydrogen ions. So the thylakoid membrane ensures that there is a buildup or accumulation of hydrogen ions. These hydrogen ions can only leave the thylakoid lumen via the ATP synthase. This process is referred to as chemiosmosis. So just a quick pause here. So this whole chemiosmosis process also happens in the mitochondria. I'm just going to draw out a little mitochondria here. So that's the outer membrane, and what I'm drawing here is the inner membrane. And then what happens is they're pumping protons to the intermembrane space. Right, and again, you have that buildup of hydrogen ions, and the only way they can leave is through the ATP synthase. So it's the same process that we see in the mitochondria as well. Let's go over the marks. So the first mark is contain photosystems that absorb light. So contain photosystems that absorb light. That's one mark. The second mark is electron transport chain and pumping protons. So electron transport chain, which is used to pump protons. That's the second mark. The third mark is allows for the membrane, allows for the accumulation of these protons or hydrogen ions. That's the third mark. And then we can say that ATP synthase allows for the hydrogen ions to leave. That would be the fourth mark. And the fifth mark here would be mentioning the terms like chemiosmosis, for example, or you can say this process is referred to as photophosphorylation. All right, let's go on to part C. The artificial chloroplasts made by the scientists consisted of thylakoids removed from spinach leaves and enzymes involved in carbon fixation. These components are put into water, forming a suspension. The suspension was dropped into a medium containing oil. The suspension formed cell-sized droplets of water containing the thylakoids and the enzymes. So the first part, it says, name the enzyme that fixes carbon dioxide in the light-independent stage of photosynthesis. That would be the first enzyme that we see in the Calvin cycle, and that would be rubisco. Or you can say ribulose bisphosphate carboxylase. That's actually the short form for that term. All right, part 2 Explain why it is necessary to keep the thylakoids and enzymes together within droplets. So let me show you this diagram here. So the light photosynthesis consists of two reactions, your light dependent and your light independent. Your light dependent takes place in the thylakoid, and your light independent involves all of these enzymes like ribisco, and that will be your Calvin cycle. So it's important to keep these two close together because the products, you see here, the products in red that are used in the light-dependent reaction is used as reactants in the light-independent reaction. And likewise, the products of the light-independent reaction are used as reactants for the light-dependent reaction. So that's why these two need to be kept close together. So we can write, it is important to keep the thylakoids and enzymes close together because the products of the light dependent stage is required for, say, carbon fixation or the light independent stage. And vice versa. Another mark you can also say is because they're within droplets, so you can say that the enzymes need to be in solution so that they are able to move around and collide with its substrate. All right, let's move on to question 7. The Yap Trench is an area of very deep water in the Pacific Ocean. In 2017, a new species of fish was caught in the Yap Trench. This was named the Yap Haddle snailfish. The photograph shows a different species of snailfish, the Mariana Haddle snailfish. So it says the Yap Haddle snailfish was caught in a deep sea trench at a depth of 7,000 meters below sea level. Hydrostatic pressure increases with 100 kilopascals every 10 meters of depth. Calculate the hydrostatic pressure at 7,000 meters. All right, so how many 10 meters can go into 7,000? So we would do 7,000 divided by 10, and then times it by 100 kilopascals. And that would be 70,000. And then to give the answer in standard form, this is what I personally do. I'm going to write this out in orange. So imagine the decimal point here. Oops. The decimal point is somewhere here. And I'm going to move it 1, 2, 3, 4 spaces. Therefore, that would be 7 times 10 to the power of 4. 7 times 10 to the power of 4. All right, the analysis of the genome of the Yap Haddle snailfish and the Mariana Haddle snailfish showed that they were closely related. The DNA from the Yap Haddle snailfish contained this many bases, and of those, these ones were adenine. Calculate the number of guanine bases in the DNA of this fish. So, if you recall, adenine pairs up with thymine and cytosine pairs up with guanine. So if you have, let's say, 100 of your adenine, you would have 100 of your thymine, right? So let me just write this out here. C and G. Oh, one way to remember this is that the letter A and the letter T are both pointy, so they pair up with each other. And the letter C and the letter G are curvy, so they pair up with each other. Okay, So we have 204, 202, 736 for actually both of these bases, for both adenine and thymine. So we can write that out as, okay, this is going to be a very ugly equation. So 725608564. So that should equal 2 times of this number, because you would have this amount of number for adenine and this amount of number for thymine. So we would write 2 times 204, 202, 736. And this, let's assign X as a number of cytosine bases, and therefore the number of guanine bases would also be X as well. So that would be plus 2X. So let's solve for that. So that would be 317,203,092. And let's divide that by 2. So we should get 158,601,546. All right. 1,546. Okay, let's look at part 2. The Venn diagram shows unique and shared gene families in the genomes of 3 species of snailfish and a zebrafish. Calculate the percentage of the gene families in the Yap-Hadal snailfish that are shared with the Mariana-Hadal snailfish. Give your answer to 1 decimal place. So I'm just going to highlight some of this so you can see. So with the Yap-Hadal snailfish, we have this whole lot here. And then let me do this with another highlighter. With the Mariana-Hadal snailfish, sorry about the pronunciation, we would have this. Okay. So what I'm going to do is I'm just going to calculate the number of genes in both, in this one and in this one, and then also in both. So let me just calculate that first. Okay, so I've calculated the number of genes found in the Mariana-Hadal snailfish by adding these 1, 2, 3, 4, 5, 6, 7, 8 numbers together. And then I've also calculated the number of genes for this species. And then for both, I would add up these 4 numbers together. And these are the values that I get. So for this question, there's 2 ways to go about this. Either I can do this number divided by this number times 100, or I can do this number divided by this number times 100. I'm just going to opt for the first option. So let's write that down. So 12658, divide that by 14750. And we're going to times that by 100 and make sure that your answer is in 1 decimal place, or to 1 decimal place. So let me just type that in the calculator. And we should get 85.8%. All right, 85.8%. Okay, let's look at the next part. The diagram shows the evolutionary relationship between 3 species of snailfish and a zebrafish. Okay, analyze the diagram to describe the evolutionary relationships between these 4 species of fish. Okay, let's scroll back up. So just to give you a little bit of background about this type of diagram, this is referred to as a cladogram, and it showcases the evolutionary relationships between species. So here we refer to as the root. This is the common ancestor. And every time you see this branching off, right here, here, these are what we refer to as nodes. Oops. So these are nodes. And this is basically, a node would represent sort of this hypothetical speciation event in which the common ancestor, you can see, splits into 2 groups, right, or 2 species. So the more nodes you have between two groups, the more distantly related they are. And the fewer number of nodes you have between two groups, the more closely related the two species would be. So if we look here to the left, we can see that all four species share the same common ancestor that existed about 230 or 240 million years ago. And then that's when they diverged into two groups. Right, so that would make the zebrafish more distantly related compared to the three of them. Now, in terms of which species are more closely related, we know that the yap haddle and the Mariana haddle are both very closely related, or I should say the most closely related, because they have the most recent common ancestor here. So let's write out some of these points. So from the diagram, we can see that, that all four species came from the same common ancestor. The zebrafish are the most distantly related. Diverged 230 to 240 million years ago. We can also say the yap adult and the Mariana haddle snailfish are the most closely related. As they have the most recent common ancestor. All right, let's go over the marks. The first mark is that all four species came from the same common ancestor. You can say another mark where the zebrafish are the most distantly related because they diverged about 240 million years ago. So that's the second mark. The third mark is acknowledging that the yap haddle and the Mariana haddle snailfish are the most closely related. That's a third mark. And that's because they have the most recent common ancestor. That would be a fourth mark. Another thing that you can also say is the yap haddle, let me just circle this. The yap haddle and the Mariana haddle both share a common ancestor with the Tucana's snailfish as well. Okay, let's go on to part 4. The Mariana haddle snailfish and the yap haddle snailfish were caught in separate trenches, hundreds of kilometers apart. Explain how these fish evolved to become separate species. All right, so if you recall, there are two modes of speciation. We have allopatric, which is the top one here, and then we have sympatric, which is here. So allopatric is when the speciation event occurs in two different regions, whereas sympatric is when they occur at the same region, which is at the bottom here. In this case, because the two snailfish were caught at separate trenches hundreds of kilometers apart, that would indicate that it's the top one here, which is your allopatric speciation. So I'm just going to remove the diagram at the bottom. So what happens in allopatric speciation is you have the common ancestor that lived in the same area. And then a geographical barrier occurs. In this case, this geographical barrier created these separate trenches, right? And because of that, these two regions now experience different selection pressures. So maybe the food that is available there is different, or the predators that are present in the two regions are different, right? And because the selection pressures are different, what the traits that allow them to survive would be different. And then that's where we talk about natural selection. So for natural selection to occur, you need to have variation. So for example, if we're talking about the fish, maybe some snailfish are larger than others, some snailfish have a different color than others, and these are variation that can only arise due to mutations. For example, perhaps there might be a certain skin pattern or a certain color of their skin that enables them to camouflage better and hide from predators better. And then this would be a trait that would increase their survivability. So they're more likely to survive, they're more likely to reproduce, and then they can pass on their alleles to their offspring. So that over time, you have more and more of these alleles over generations. So just to recap first, you would talk about number one, there is a geographical barrier, and that causes these two populations to become isolated from each other. Number two, you talk about how these two populations would experience different selection pressures. And number three, you talk about natural selection and how the allele that enables them to survive better would then get passed on from one generation to the next. And that overall increases their allele frequency. And again, because of this geographical barrier, there is no gene flow that is occurring between these populations. And eventually, all of these differences accumulate with each other until they become reproductively isolated, which means that they are no longer able to interbreed to produce a fertile offspring. And that would show that, now they're two different species. Another aspect that's mentioned in the marking scheme is epigenetics. You just write that down. So if you recall, these are changes that are made that affect the transcription and the translation of the genes. So essentially, it causes the genes to be switched on or switched off. And these epigenetic modifications can be inherited. So you can also talk about that. I looked at the examiner report, and typically students don't mention epigenetics, and that is fine. You can still get the 6 marks by mentioning the first 4 points that we see here. So when you look at the marking scheme, we can see that in order for you to get 6 marks, you need to be able to properly explain 3 of the aspects plus 1 aspect and include a specific detail related to that. So that is how you would get the 6 marks. Okay, I'm just going to erase this. This would be an example of allopatric speciation. The common ancestor of these two snailfish lived in the same region, but were then separated by a geographical barrier, causing the formation of the separate trenches. These two populations would experience different selection pressures. For example, there may be different predators in these trenches. These populations would have experienced natural selection. For natural selection to occur, mutations must have given rise to variation. There may be certain phenotypes that would increase their ability to survive. So, therefore, they are more likely on their advantageous allele to their offspring. Over time, this increases the allele frequencies. And then lastly, we talk about the gene flow or the lack of gene flow. That there is no interbreeding between the populations, which means no gene flow occurs. Eventually, they will accumulate enough genetic differences that they become reproductively isolated. Okay, that's a lot that I've written here. Let's go over the sections first. So again, this first section here talks about isolation. This part here talks about how they would experience different different selection pressures, This part here talks about what is natural selection. And this part here talks about how there's no gene flow that occurs between them and eventually they become reproductively isolated. And that's it for part 2. For questions 8 and 9, I will be going over that in part 3. So just click on the link in the description below. Bye!